Hello, everyone, and welcome to the TrialSite News Podcast, Farago. I am Erin, your host. Thank you so much to everyone for joining in. And today we are joined by Dr. Jake Scott, and he is an immunologist at Stanford University. And before we dive into today's topic, Dr. Scott, thank you so much for being here. And do you mind telling our viewers a little bit about yourself and what you do?
Yeah, my pleasure. And just to clarify, it's a common sort of point of confusion, but I'm not technically an immunologist. I'm an infectious diseases doctor, but there's a lot of overlap. And I primarily see patients as an infectious diseases consultant. And I do want to just take a second to explain that, because even when I tell friends and family what I do, they're often assuming that I just see exotic diseases. I just deal with, Ebola or whatever, or COVID. But no, what I do is mostly see patients with a variety of infections from head to toe, whether it's, meningitis, pneumonia, sepsis, where the body is overwhelmed by an infection, urinary tract infections. So I primarily see patients in the hospital who are really sick with a number of infections. In 2020, that was primarily COVID. These days, it's a wide variety of stuff. And then I also see patients in my office. I specialize in HIV, so I've got a fair number of patients with HIV that I manage, a whole variety of infections. I'm also on faculty at Stanford and really enjoy the academic side of stuff. I really enjoy writing, so I write a fair number of review articles. I'm involved in some clinical trials. I'm involved in a long COVID trial right now, the RECOVER trial. And I'm also the director of the antimicrobial stewardship program at my hospital, which is basically all about just making sure that we're using antibiotics judiciously. And my passion outside of that is science communication and trying to just make sure that people have the right information or the best information so that they can make the best decisions for themselves.
Okay, perfect. So scientific communications, any other hobbies?
Yeah, I should say I don't just do that. I have two young kids, and I spend a lot of time with them. I like to go on trips and stuff. We just went to Mexico. I'm also really into rock climbing and just recently got into scuba diving. So
Nice. Okay.
yeah.
All right. Outdoorsy. Okay. All right. So just for our viewers, today we're going to talk about vaccines. I know it's a topic everyone agrees on, no, but hopefully this will be a respectful, informative conversation. There's a lot of conversations out there happening, and I think it's important to keep having respectful conversations where people can ask questions and get the information that they need to be confident in their decisions. So that said, Dr. Scott, you and a team of folks began a project in April of this year where you searched for every randomized and placebo-controlled vaccine clinical trial in the medical literature, including in other countries. So can you tell our viewers more about that?
Yeah, so it's been fascinating. So on April 22nd, one of my sort of heroes in medicine and infectious diseases and friend who I've collaborated with over the years, Brad Spellberg, he posted something on X about how it would be great if we could crowdsource data collection looking at all the controlled vaccine trials. I have to say, I've learned a ton while doing this project. And I just have to say, the science of vaccines, which we call vaccinology, and the science of clinical trials, it's very complicated. I've been focused on vaccines for a while now, several years, and I'm still learning a lot on a regular basis. And the terminology is confusing, and I say that not just because I sympathize with how confusing it must be for people who aren't trained in medicine, because I find immunology and vaccinology very complicated. And also to clarify, we're not looking at just placebo-controlled trials, although that was sort of, the original sort of impetus was to see how many placebo-controlled vaccine trials there are. We're also looking at every controlled trial in general. So whether they use an active comparator like a pre-existing vaccine, or a vaccine for an unrelated disease. So I started a Google sheet and seeded it with 7 vaccine trials that I found, and then we made it go live. I embedded the sheet on Brad Spellberg's website, which you could see, bradspellberg.com. And he's done a lot of work around antibiotic stewardship and collecting data on proving that shorter is better, so shorter courses of antibiotics. And so this is another sort of section of his website. And then it really took off. So people from around the world continue to contribute and put data into this sheet. So it went from 7 trials to over 40 or so in a day or so. And we're now at 293 controlled vaccine trials going back to the 40s. And it's funny, I sort of thought that we were done at the end of May. And I even said, something like wrapped up, data extraction. But then I went back and I've sort of been honing my PubMed searches, how to find these data. And I just keep finding more and more. And I think I've finally sort of honed a pretty efficient yet broad search. And so I'm now going decade by decade. Randomized controlled trials kind of started in the 40s, so I'm starting with the 40s. There's really nothing before then. So I'm giving my team tons more trials to look through and to screen and see if it meets the criteria, and if so, we're putting it into the sheet. So I think that we had been looking at the tip of the iceberg for a while, and so we'll probably continue with data extraction for another, I'd say probably another 2 or 3 weeks, and I imagine we'll get to 400 plus controlled vaccine trials.
Wow. May I ask who's funding it, or is this just volunteer?
Great question. Absolutely no one is funding it. This is what I do after I put my kids to bed, and
I don't. Your hobby. Okay.
Yeah, I'm kind of obsessed. I should probably sleep more, but yeah, none of us get a cent. And I also want to explain that I get a salary from Stanford because I work for Stanford and I see patients. Otherwise, I don't get any funding from pharma or any vaccine manufacturers. This is purely voluntary.
Okay, great. No, I'm so glad you said that, because that question does come up a lot.
A lot.
TrialSite News does not get any funding from pharmaceutical companies, or big pharma, as it's called, so either. Yeah. So that's good. Okay.
Yeah.
So before we dive into kind of the take-home points of these clinical trials, there has been a lot of chatter and controversy over whether or not licensed vaccines, particularly those for children, have been tested against a placebo. And I want to talk to you about the different comparative groups or the different controls that might be used. But first, can you define placebo? I like to start with the basics. So can you tell us, what is a placebo?
Yeah, great question. So when we think of placebo, I think the standard sort of instinct is that it's saline, or that it's totally inert. And inert means it doesn't trigger any sort of biological or physiological response. And the other thing I've learned while doing this is that the definitions haven't really been totally clearly established. But in general, after reviewing, FDA documents, textbooks like Plotkin's vaccines, and talking to colleagues who work in vaccinology, basically, in order to see whether or not a medication or a vaccine is effective, the best way to do it is to compare it to someone who doesn't get that vaccine. But if someone doesn't get that vaccine, then, so back in the beginning, they compared it to no intervention, right? And some people consider that to be sort of the gold standard, because then you can really see if the vaccine does anything. So say if you take 100,000 people and give them a vaccine, say for polio, and then 100,000 people and you give them nothing, which has been done. Part of the Salk trial was comparing no intervention to kids that got the vaccine. And then you'll see, whether or not there's a significant difference in mortality and disability, et cetera. But there's also something called blinding. So when a participant in a study doesn't get an intervention, a shot, for instance, or an oral vaccine, they'll know it, obviously. And so maybe they'll act or behave differently. Maybe they'll be more cautious moving forward, knowing that they didn't get the vaccine. And then the investigators would also know it. And so this leads to bias, which could skew the results of the study. So instead, scientists started using placebo, meaning that they're given something, but it's as close to nothing as possible, right? So, but it's not nothing. It can't be nothing because then they would know they got nothing. So one sort of classic example of a placebo is a saline injection. And saline is just salt water. So say you take 100,000 kids and you give them a flu vaccine, and then you take another 100,000 kids and you give them a saline placebo. Well, then you could compare the outcomes in both groups or between the groups and see whether or not there's a difference in the number of flu cases, the severity of flu, side effects, safety profiles, et cetera. One issue, though, is that in order to really optimize the immune response, vaccines are commonly given with what's called an adjuvant. And an adjuvant is something that basically nudges the immune system to do its job. And back in 1926, scientists discovered that aluminum salts, aluminum phosphate, was very effective at stimulating an immune response. So it basically helps sort of keep the target that the immune response is supposed to focus on. So say a protein particle from a virus. And the aluminum adjuvant basically provides what's called a depot. So it allows that to sort of just sit there for a little longer so the immune system can train itself on it for longer. And it leads to a number of better quality immune responses. And importantly, and I think that this is a really important point that people often don't realize, is that the aluminum adjuvants, they not only stimulate a better quality immune response, they also allow for antigen sparing. And an antigen is basically sort of, it's like the mugshot that we give to the immune system. And we say, hey, recognize this. If you see this guy or this person, it's not gender, specific, then, mount an immune response. So that's the antigen. It's the little piece of information about the infectious disease, the microbe, that allows the immune system to remember it and attack it. That's called the antigen. And aluminum adjuvants allow for there to be less antigen. So it's a major win. So aluminum adjuvant, again, provides a better quality immune response, but it also allows for the vaccine to include less of an antigen, less of the piece of the microbe. But it causes a side effect. And the side effect is local pain, swelling, redness, usually mild. But, everyone who's gotten a shot will know that, yeah, there was some soreness, some swelling, some redness. And that's primarily from the adjuvant. And it's also a sign that the immune system is working. So I think of it as kind of like, after you go to the gym, if you get a good workout, you're usually a little sore. And that's a sign that your body's, working, no pain, no gain. So that's aluminum adjuvant. Mm-hmm. And and so if we gave kids a flu shot with aluminum adjuvant and then saline in the control group, and the control group just means that's who we're comparing it to. Well, then every kid in the control group, if they just got saline and they didn't get the redness, the swelling, the pain, then they would know, and their parents would know that they got placebo. So we started giving them, not me, but scientists started giving them aluminum adjuvants as placebo without the flu antigen. And flu is just one example of many. So we still consider that a placebo because it's what we call immunologically inert. So although the aluminum in the adjuvant allows for a better quality immune response, the immune system doesn't end up targeting the aluminum. The aluminum gets processed and cleared by the kidneys and has been proven to be perfectly safe after 70 plus years of data. So anyway, long story short, there's saline placebo, but then we also use aluminum adjuvant and other adjuvants and consider those placebo as well. And they're not 100% biologically inert, but they're immunologically inert. And so to say that, no vaccine was placebo controlled, well, that's just not true because a lot of them have been, especially with the childhood vaccines, tested against saline placebo, but a lot have also been tested with aluminum adjuvant placebo.
Okay. Let me just jump in there. So our viewers understand. So we have the inert placebos, which is saline. We have the adjuvant only placebos,
Exactly.
which is like the assistant in there, and then we have the active comparators, which might be another vaccine. Do you want to talk a little about that? Okay.
Yes. Yeah. So in general, I consider all of these to be controls. So what you're comparing the study group to, right? So controls could be either no intervention, which sometimes we do, but not that often. But there's still some recent studies that have looked at that. And then there's saline placebo, and exactly like you said, and this is how we categorize it in our sheet. And then adjuvant placebo, and then the others we don't. Well, and then there's excipient only placebo. So sometimes vaccines have to be given with what we call stabilizer. So basically just to make sure that the particle that the immune response needs to target it remains stable and doesn't sort of decompose or whatever. And those are benign. And so we call those excipients. So there's saline placebo, adjuvant placebo, excipient placebo, at least in our terminology. And then there are active comparisons. So if you give somebody a new and improved pneumococcal vaccine, and I just want to say the pneumococcal vaccine is often misunderstood as the pneumonia shot. And it's not the pneumonia shot. That's a misnomer. Pneumonia just refers to a lung infection caused by a wide variety of things. I mean, pneumonia can be caused by viruses, bacteria, fungi, and the pneumonia shot is not an accurate term. It's the pneumococcal shot, which refers to an immunization against Streptococcus pneumonia. And Streptococcus pneumonia was called that because it was the most common cause of pneumonia. So anyway, I digress a bit, but then say you want to study a new and improved pneumococcal vaccine. Well, if we know that it provides safe and effective protection against invasive pneumococcal disease, which can cause really bad sepsis and meningitis, etc., then it wouldn't be ethical to give some kids the real vaccine and some kids placebo, right? Because you can't withhold this vaccine that has already been proven to be safe and effective. And so that's often the case these days. As we have more and more vaccines available that are proven to be safe and effective, we can't study new and improved versions with placebo. So we have to give them the available vaccine or the already licensed and marketed vaccine.
And just to go back, when you said sepsis, can you just explain to our viewers what that is, just so people
Yeah, great question,
will
because
know?
that's all, yeah, that can be a confusing term. Sepsis refers to when the body is overwhelmed by an infection, and any number of infections can do it, but it leads to, or sepsis describes when the body has this, pretty dramatic inflammatory response, and it can lead to fevers, high heart rate, low blood pressure, et cetera. And there's a spectrum, but ultimately, the extreme end of sepsis would be septic shock, where you're in the ICU and you're on medications that increase your blood pressure, et cetera.
All right.
It's very serious. It's one of the most common causes of death, and
Right, right. But I appreciate you talk because we're talking about vaccines and we're hyper focusing on this, and then I always have to remind myself that part of this conversation is what are you vaccinating against, right? Like when we're doing those risks benefits, considerations.
Totally.
So,
Yep.
so let's get into the trials. Can you tell us, how many you went through, and of those, how many were tested against an inert placebo versus adjuvant only, active comparators? Can you just give our viewers a sense of those numbers?
Yeah, and I have that right here. Let me just also add the last category would be an active comparison with a different vaccine, a vaccine for an unrelated disease. And so say you're giving someone a new vaccine, say against, hepatitis B or Neisseria meningitidis, and you're giving it to a population that doesn't have access, say it's in, another part of the world that doesn't have access to a rabies vaccine. Instead of giving that control group nothing, you could give them some vaccine that provides some protection against different disease, just so that you're not leaving, a control group totally hanging. So anyway, that's the last group.
Thanks.
Yeah, so I'm looking at the sheet now, and as of June 16th at 10 a.m., there are 294 controlled vaccine trials. And these were vetted by my team and myself. And so I'll do a massive PubMed sweep and find hundreds and hundreds, sometimes thousands of trials. And then I'll filter them out just to make sure that they meet our criteria, meaning that they're randomized, that they're not just observational, that they're controlled, there's a control group, and that they involve humans, not animals, et cetera. And so we start with casting a wide net, and then we read through the abstracts and the papers. And at this point, there are 294 trials. And the trials, we also input the number of participants in the studies, in the vaccination group and the control group. As of now, the live total number of trial participants is 3,185,780. The placebo and inert trials add up at this point to 138. The caveat is that this is a work in progress. I know that my team, my colleagues put a number of new trials in there, and I think for some of them, the placebo wasn't totally clear, so we've got to hunt down some of the papers. So that'll change.
Okay.
And then the total number of trials that used placebo adjuvant, 35. Okay. And then active control, same disease vaccine, 44. Active control, unrelated disease, 52. Other, 8. No intervention, 10. Okay. So, so the inert placebo, again, that's primarily saline. That's not the only one, though. So sometimes, an oral buffer placebo, basically just salt water. So another one would be like liquid calcium carbonate, which they use for rotavirus placebo. But yeah, 138 inert placebo trials out of 294. So, and that's been pretty steady throughout our search. About 40 to 50% of the trials have been inert placebo controlled.
Now, there's been a lot of discussion about the childhood vaccines that are currently recommended by the CDC. So I wanted to ask you specifically about those. Can you talk a little bit about what those approved vaccines were tested against in these trials? From the ones you found. I know you said you're still looking, you might add more, that kind of thing.
Yeah, yeah. So, there have been a number of trials for the childhood vaccines. Let me pull up, because I don't want to misspeak.
Sure.
Let me pull something up in particular. One sec.
I didn't mean to put you on the spot with
No,
specific
no,
numbers.
it's fine.
But...
Yeah, no,
okay.
this is a very important question, and so I'm ready. I just want to make sure
Okay.
that it's totally clear.
Good.
Okay, so the childhood vaccine schedule for kids these days shows 16 or so different antigens. So an antigen for polio, rotavirus, etc. Okay, so of those, every single one has been studied with a placebo of some sort, whether that's saline or some other salt or aluminum adjuvant. So I do consider aluminum adjuvant a placebo. And I do, one of the reasons why we separated that in our sheet was because, people want to see which placebo was, totally inert, biologically and immunologically, and then which placebo was, aluminum adjuvant, and there are other adjuvants as well, but those are what we call immunologically inert, but not biologically inert because they are biologically active, but still safe. So the polio vaccine, that was one of the largest prospective clinical trials ever. It was massive. There were 200,000 kids that got the polio vaccine. This was the famous Salk field trial of 1954. 200,000 got the polio vaccine and 200,000 got saline. But then there was a no intervention group of like 1 million kids. And there was another trial, or another arm of the study, I believe that was in Russia, that included like 1.8 million kids. And, and their outcomes were all, monitored. So massive, massive trial. So placebo definitely checks the box there for placebo control. But polio checks the box for placebo control.
Okay.
Rotavirus, massive, massive amount of data, 12 inert placebo randomized control trials, including the REST trial, which had 70,000 kids. I myself looked through a lot of this data because I've been getting this question a lot from folks in the media. And, there's some that are a little harder to find, but I did find a diphtheria saline controlled randomized trial, and that's on the vaccine schedule. That was a trial that looked at both Haemophilus influenza and diphtheria. So I haven't found a solo diphtheria or diphtheria only vaccine that was saline controlled. But
just so can you say how they would recognize that vaccine?
Oh yeah, so thank you. So that's typically given as Tdap or DTaP. So with tetanus and acellular pertussis.
Right.
And so that's the D and DTaP, or the D and Tdap. Right. And then tetanus. There was a distilled water control trial. Pertussis, which is in the Tdap. There were 2 no intervention randomized control trials. So kids who got pertussis vaccine and kids who got no vaccine. And when it comes to how inert a control group is, I mean, you don't get more inert than no intervention, right? Because they didn't even get a shot. They got nothing. And then pneumococcal vaccines, there were multiple saline control trials. Measles, mumps, rubella, there was an antigen-free diluent trial, and the diluent is what basically the vaccine is just sort of dissolved in. So there is that. There was an HPV saline controlled randomized control trial. HPV is human papillomavirus, which is a common cause of cervical cancer. So this vaccine not only prevents infection, but dramatically lowers the risk of cervical cancer, which is a major win. Hepatitis B, there were two aluminum adjuvant control trials. So yeah, there are a lot.
Okay. So let's address some of the things that I'm sure we both have seen floating around online that people are concerned about. So this concept of inert placebo versus adjuvant only placebo. And there's a lot of concern over how safe a vaccine is. As you know, we give these to babies and children. So obviously it makes sense to be concerned about safety. If a placebo group in a vaccine trial is given an adjuvant containing placebo, in other words, as you established before, a biologically active substance, although you said immunologically inactive, how can someone be sure that it will not mask safety signals for the vaccine being tested?
Yeah, so I mean, one example is aluminum adjuvant, and not to beat a dead horse, but that's far and away the most studied. There are more types of adjuvants, but aluminum adjuvants have been extremely extensively studied, and so and I told you about the you know, why aluminum is there and what it does. It's basically an immune helper, right? But the aluminum adjuvants have been part of routine vaccines since the 30s or so, and billions of doses have been given, and there have been decades of monitoring worldwide by not only the CDC but also the European Regulatory Agency, the World Health Organization, and they all consider them to be safe. And so the other thing is the dose, the amount is extremely small. So a typical vaccine contains 0.2 to 0.5 milligrams of aluminum. They're not allowed to include more than 0.85 milligrams. And we typically consume 5 to 10 milligrams every day from our food and water. And yeah, there is certainly a difference between the amount of aluminum that we ingest and the injection, but they both ultimately get into the bloodstream. When we ingest anything, it gets absorbed into the bloodstream. And when we get a shot, it ultimately gets, goes through the bloodstream. But then it gets filtered by the kidney and then ends up in your urine, and some of it ends up in the stool. So the dose that we get in the injection is so much smaller, thousands of times smaller than what we consume on a dietary intake. Does that answer your question?
Yeah, I think this is just a question that I've seen floating around. So I thought this was a great opportunity to address that concern. People, just because it's not just pure saline, right? And I'm sure you've seen this if you're active on social media, that it's a concern.
Yeah, and I completely understand why it's a concern. I do. And there's been a lot out there about adjuvants and aluminum adjuvants in particular. But vaccines have been, I would say, the most extensively studied medicines of all time. They've I mean, like I said, we're looking at the tip of the iceberg here with nearly 300 controlled vaccine trials. I think we'll get to over 400. And the vast majority, over 90% of those trials included safety outcome data. But the other thing is, people say, well, you can't necessarily detect a safety signal. You can't you can't necessarily find, a rare serious side effect in a randomized controlled trial. And that's true. And that's why we have post-licensure monitoring.
Right.
A number of different programs.
I have some
And
questions about that for you down the road.
yeah, yeah, and so and those have been very effective at picking up rare but serious side effects. So with the Johnson and Johnson COVID vaccine,
Mm-hmm.
we picked up rates of blood clots, and
Sure.
then it was pooled. With rotavirus, after the randomized clinical trial, we picked up rates of intussusception, which is where the intestine kind of folds in on itself,
Right.
and then that was taken off the market. There was an RSV trial in the 1960s that during the trial looked okay, and then soon afterwards, there were adverse effects, including a couple of deaths. And that's because it had enhanced disease, which is kind of immunologically complicated. But the other thing I want to say about the aluminum adjuvant question is that one common concern, and I just posted this on X recently,
Okay.
or this morning, is that kids are getting too many vaccines, and I can totally understand why that would be a concern. I hate giving my kids anything that they don't need, whether it's a shot or a pill or whatever. Um, and yeah, nobody wants to give their kids too many vaccines if they're not, totally necessary, right? And a big part of my job is, talking doctors out of using antibiotics, for instance, because I'm into antibiotic stewardship. But one thing I found really interesting when looking through some data is that the overall amount of antigens is far less than it used to be. And so again, antigens are the piece of virus or bacteria, which are typically proteins or sometimes sugars, carbohydrates, that teach your immune system to recognize and respond to an infection, right? So, so the idea that we're giving kids too many vaccines, well, I don't think that really holds water, because even though kids these days get about 50 injections by the age of 18, the total number of antigens is far lower than it was in 1986, for instance. So in 1986, kids got about 10 vaccines or 10 injections, and then polio, which was oral. But those vaccines were different. They weren't sort of as they were broader. They include whole organisms or whole cell vaccines. So those 10 shots over 18 years exposed kids to about 3,000 antigens. These days, 50 vaccines expose kids to about 150 antigens.
Okay.
So that's a huge reduction. And then the aluminum adjuvant also has really hardly gone up at all in total. So it used to be about, the total exposure over 18 years was about 3 milligrams in 1986. And now it's about 5 to 6. But the amount of aluminum adjuvant per vaccine these days, per dose, is smaller. So we're giving a lot more. There's a lot more bang for the buck. So I just want to point that out, because it's
I
commonly.
appreciate that. Another concern, adjuvants are not inert. We have established that. And some people are worried about perhaps long-lasting effects on the immune system. We heard of autoimmunity, or, where the immune system might attack your own body or something like that. And people see that there may be an uptick in those types of disorders. Is there a possibility at all that we're not studying them enough, that's a possibility? Can you address that?
Yeah, great question. This has been studied, and what's interesting is that, again, we're talking about one, exposure to antigens, two, exposure to adjuvants, right? When it comes to antigens, what we're seeing on a daily basis when we're not getting vaccines, in terms of what we eat or breathe in, our immune system is seeing a massive amount of antigens on a daily basis. And so we get exposed to far more antigens from a single cold or just a walk outside than we do from those 150 antigens spread out over 18 years with vaccines. And that's just how the immune system works. The immune system is extremely complex and adaptive and resilient, and and it sees a lot more antigens than it sees because of vaccinations. So there's this very common misconception that the immune system is delicate and that too many vaccines will disrupt it or lead to dysregulation or what someone said deregulation, which is not the scientific term. But our immune systems have evolved over millions and millions of years to adapt to all these different complex exposures. And there's no evidence, despite extensive studies, that these vaccines have led to any sort of immune disruption. There might be some role with immune, an abnormal immune response that then leads to a rare side effect. So, myocarditis with COVID vaccines. Could there be some sort of, autoimmune component? Perhaps. But on a large scale, this has been studied a lot, and no, there's really no evidence that it leads to overwhelming the immune system or autoimmune disease. And again, that's also been studied with aluminum adjuvants extensively, and no evidence.
Okay. Can we talk about active comparators? You mentioned, you explained before why older vaccines might be used. It would be unethical. Probably the folks who approve a research trial would not approve it if there was a licensed vaccine, out there. They would say we can't give that, those folks a placebo because they would be without protection. It would be unethical. However, sometimes that some people argue that may make a vaccine look safer than it really is because side effects are more likely in both groups. Do you think that is a valid concern?
That's a good question. Yeah. Well, just to back up, so an active comparator means that the control group receives a different vaccine, right? Usually one that's already licensed. And it's used when given a placebo would be unethical because there's an effective vaccine that exists. So the pneumococcal vaccine, like I said, is an important example. And so the other thing is a lot of these vaccines had already been tested against placebo, right? So when a vaccine is totally new, when there's no existing vaccine for that particular infectious disease, it's usually primarily studied initially against placebo or aluminum adjuvant or some other adjuvant. And then we continue to improve upon some of these vaccines. So, there are multiple different pneumococcal vaccines that are sort of new and improved. And those are compared to pre-existing or already available vaccines. I don't see why overall that would lead to a skewing of the safety data and not, not reveal any concerning safety signal unique to that vaccine. But I can understand the question and the concern.
Okay. Another concern, we give vaccines to healthy people. And what I mean by that, I mean people who aren't ill with whatever it is you're vaccinating against. So if we're designing trials for healthy populations, especially children or pregnant people, although I know there's not a lot of trials in that population. Should not the default be the most transparent, conservative approach to placebo choice unless there's a compelling reason otherwise? What are your thoughts on that?
No, I agree. So I think that when it's possible, yes, the default would be the most the safest, most inert control. Does that make sense?
Yeah, yeah.
Okay.
I'm just taking concerns that I've seen and throwing them at you
Yeah, no, I
because
appreciate this.
you're the
These are
expert.
great. These are great questions, and I also want to emphasize to the audience that I totally value skepticism and critical thinking. I do not want to push anything on someone, especially if they don't understand or if they have any questions. So I think all of these questions are great, and I encourage people to ask
Okay.
whatever question comes to mind. So
Okay.
these are great.
And I also want to emphasize, while I'm, throwing questions about vaccines at you, the potential risk of a vaccine obviously does not exist in a vacuum. There's always the other side of the equation, the threat that the disease itself poses that often gets lost in these conversations. As a scientist and a clinician, how do you think through that risk-benefit balance? And just as importantly, how do you help your patients or the public navigate that, especially since science is still evolving and especially at a time when public trust is fairly low?
Yeah, great question. I mean, I feel like a big part of my job as a physician and clinician is to take the time to go through the risks, the benefits, and the alternatives, and to give people the evidence that I know. So give them the best information I can, the most accurate, evidence-based information, so that they can make informed decisions to make the best decisions for themselves. And so I believe in shared decision making. And on the other hand, I do believe that there's a very important role for guidelines that are based on data and expert input. So we need both. And so, yeah, it's whenever we're talking about any medicine, whether it's ibuprofen or an antibiotic or a vaccine, it always comes down to weighing the risks versus the benefits and considering the alternatives, right? And the other thing I'll say is that thanks to vaccines, there are so many diseases these days that really aren't an issue. But if it weren't for vaccines, they would be an issue. So it's hard to sort of acknowledge that there's a risk for influenza B meningitis when there really isn't much of that anymore, thanks to the availability of that vaccine. So, if and I'm a parent, so if my kid's pediatrician is recommending this vaccine, and I think to myself, well, my kid's not going to get that. My kid's not going to get Hep B or Haemophilus influenza B, and I'm not being serious. I'm just saying hypothetically. Well, that's because of the vaccines. It's not, so it's a real sort of luxury that we have, that the risk is so low. But you got to compare that to the relative risk from the vaccine. The risk from the vaccine is extremely low for the vaccines that are widely available. And so the benefits do outweigh the risks. There are other cases where I don't think the benefits necessarily outweigh the risks, even when the risks are very small. Sometimes the benefits are really, really small. And I'll be candid with patients about that. And if they don't want to take that chance, then, or if they want to just, not get that vaccine, that's fine.
So joint decision making, it sounds like.
Totally, yeah.
Okay. Do you have time for a few more questions?
Sure.
Okay,
Yeah.
okay. I wanted to switch gears a little bit and talk about the vaccine adverse event surveillance system. Most of our listeners know it as VAERS. It's a passive surveillance system for detecting adverse events related to vaccinations. And what I mean by passive is that people have to report into it. And while these systems have their issues, they are necessary for detecting rare adverse events that would not get picked up in a clinical trial, simply because the trial is too small. There's not enough people. And you can kind of think of it as a smoke detector. They're sensitive and helpful, but not every beep means they're a fire, and not every fire gets picked up. But it just helps scientists figure out what might be happening. Dr. Scott, what are your thoughts on how well VAERS works right now?
Great question, and I love that analogy. Yeah, I mean, there's a lot of noise in the VAERS data, but it can also be totally life-saving. Like you said, with any clinical trial, we can't enroll 1 million people in a COVID trial, a COVID vaccine trial, or whatever. So the trials are often in the thousands, and if there's a side effect that occurs that's rare, say it's 1 in 1 million, then yeah, that's not going to be picked up in a trial of thousands. So it's got to be picked up after licensure. And that's kind of the norm, with vaccines. Sure, we look at the vaccine trials, look at safety outcomes, and they monitor all that thoroughly, and they report all of that. But that's obviously not going to be enough if there's a side effect that occurs, later after the vaccine trial ended, or if it's just so rare. So for that reason, the post-licensure or post-marketing databases or systems like VAERS are absolutely essential. Again, the Johnson & Johnson vaccine is a perfect example where blood clots were found with VAERS, and then and the vaccine was taken off the market.
Right, that was an example of it working.
Yeah, and there are a number of other ones too. Yeah, the other thing I just want to say that I forgot to mention is that oftentimes when people think about the side effects, they'll think, I was just having a conversation with my dad about this yesterday. They'll think, oh, the vaccine made me sick. Well, it's the again, the sign of the immune response working. So that's what we call immunogenicity. So vaccines will often make you feel like you're ill, fatigued, flu-like symptoms, etc. And some vaccines are more immunogenic than others. COVID vaccines in particular, COVID boosters, and the shingles vaccine. But they're not making you sick. Those are, again, signs of your immune system doing its thing. And so oftentimes when we are actually ill with an infectious disease, what we're feeling, the symptoms are not a direct response of that microbe doing its thing. Our symptoms are typically a sign of they're due to the immune response. And so that's the same thing with vaccines. So vaccines don't make you ill in terms of being infected and being sick in that sense. They make you feel lousy sometimes because of your immune system doing its job.
Yeah, no, I appreciate that. I'm sure our listeners do too. I would ask, what were your views on the COVID vaccine causing myocarditis in certain age groups? Obviously, it was a risk factor that we determined during the pandemic, and a lot of folks were like, kids are, low risk, right, for COVID, and why would we risk giving them myocarditis? What would you say to a person who came into your office and said that?
Yeah, I would totally sympathize, to be honest. And this might be a little controversial, but I just have to be totally candid. So I was on the front lines and saw a ton of people die from COVID. Mostly older folks who came in from nursing homes, and nursing home outbreaks were awful. And in 2020, it was an absolute nightmare. I did see some people who succumbed to severe and even fatal COVID who were relatively healthy, and relatively young, 30s, 40s. Most of them had some underlying risk factor, but not all of them, But that was 2020. Once the vaccines became available, those death rates plummeted. I mean, I went from seeing, a lot of people dying December, January, to hardly anyone other than unvaccinated folks, unfortunately, which was totally, totally tragic. But yeah, so, and I digress a bit, but when it comes to COVID vaccines and younger people, say adolescents, and people in their 20s to 30s, I do believe that the primary vaccine series was a good thing. The primary vaccine meaning the first 2 to 3 doses. Back when the third dose was being recommended, I actually pushed back a bit and said, the primary vaccine, the first 2 doses is actually doing a very good job. And I agreed with Paul Offit at the time, and I was one of the few. I wrote a couple of op-eds about this. The booster thing, I think, got a little out of hand. And then when we clearly saw cases of myocarditis related to COVID vaccines. I might have had a few cases that were not totally clear, but I had colleagues that told me about their cases, and I've reviewed the data, and it's real. So in that sense, again, it comes to this calculus of weighing the risks versus the benefits. I've got a son, he's only 7, but if he were 18, because that's kind of when, kids are at risk for myocarditis. If he were 18, would I get him the COVID booster at this point? I mean, he's already had COVID. He's gotten, actually, he didn't even get a COVID vaccine. He got COVID. And so after that, I was like, no, I don't think that he needs a booster, and certainly not if he was 18 and healthy, because there is that risk. So, and that's me. I mean, I'm not speaking for my employer or for anyone else, but these are the kinds of conversations that I have with patients, and I often approach them as if, what would I do? What would I tell my dad or my friend, or how would I approach it? So yes, there is that risk of myocarditis, and and I do think that should have been taken seriously, and it was, but I don't mean to dwell on this, but it's so important to not hide any of these safety issues. It's all about transparency. If you want people to trust you, be honest, be transparent. Right. The data are out there. Don't pretend that something is safer than it is. It's, and that being said, of course, there are always caveats. Yes, the risk is very, very rare. But so is the risk these days of COVID-related complications in an 18-year-old.
Right. But that's a great segue to my next question. There are people who believe that they experienced or they have a documented serious side effect from a vaccine, and they often feel dismissed. They feel gaslit by the system. They're told it's a coincidence. They're told, an anecdote is misinformation, which, you can't prove it. It's not statistically significant. How should public health systems respond to those people, not just scientifically, but ethically and emotionally? What is our responsibility to them?
Oh boy, that's a great question. I mean, it's, as with any sort of relationship in life, it's so important to feel understood, I mean, that's just, I think, the crux of social relations in general. Nobody wants to feel dismissed or blown off, I personally, I hate that. And so if I tell my doctor something, I want them to take it seriously, even if it seems far-fetched. And that's how I approach my patients. Yeah, if someone has concerns that, the vaccine led to hair thinning or, itchy rash or neuropathy, you got to take that seriously. You got to give people the benefit of the doubt. And so a doctor who hears those concerns, yeah, don't blow them off. Report it to VAERS. It's easy. I've reported things to VAERS. And that's how we need more data, and so sure, we say anecdote is not, the plural of anecdotes is not data or whatever, but it is important to gather all of that information and to take everything seriously, but of course to put everything into context. Context really matters.
Right. And I always say the plural of anecdote is anecdotes.
Right.
Just leave it at that. And
yes, I like that.
They do what they do, and there's certain things they don't do, but they're not nothing.
Exactly.
Yeah.
Trust people. It's just.
Yeah. Well, and I think too, since a lot of this discourse has moved online, and I do a lot of work in scientific communications, public health communications. I think people really need to think about that environment and how they're communicating, ego, empathy, right? Like we've all felt that urge to attack if we're being personally attacked, but what are we doing? We're creating this space that is the town market, so to speak, where we go to talk about these things now.
Right. Yeah. And I think, people are understandably, I think, sensitive to being sort of talked down to, and medicine has been so paternalistic for so long, and it's so important to just like I said, give people the best information that you have access to so that they can make well-informed decisions for themselves. Don't judge, don't prejudge, and give them the benefit of the doubt and try to be understanding. So it's pretty straightforward. It should be.
It should be. Humans are complicated though, right? When the emotions and ego get involved. I want to go back to VAERS. Do you think VAERS needs any help? In your opinion, if you were in charge, what would a modern, trustworthy vaccine safety surveillance system look like if it was optimal? Would it still rely on passive reporting? Would we create something more real-time, integrated, and transparent? Do you think we need to do anything improvement-wise?
Great question. Yeah, there's always room for improvement. I haven't thought about sort of the ways in which it could be improved, but yeah, we need to invest more in safety monitoring for everything that we do. There's so many great technological tools, AI, of course, and so yeah, I think that it'll be very important to modernize VAERS. I think that the concept, is great, and the principle. Yeah. But sure, I imagine there are ways in which it could be
make it a little more user friendly.
Yeah.
Yeah, for sure.
Yeah, although it gets tricky. I mean, if you just let anybody just say, "Yeah, I got the flu vaccine," and and it led to my blurry vision. I mean. Sure, it might have been the flu vaccine, but maybe it was because your vision just got worse. So you gotta strike a balance. You don't want it to be too skewed. You don't want the data to be garbage.
Right. No,
that's not that self-reporting is inherently garbage, but there's you run the risk of
No, you do, but it's all we have. Like it's all people have right now. Like it's kind of like wait. Where do you report this? VAERS. So it shouldn't be mocked because what else is there,
Yeah. It's tricky though. It's very tricky. Someone smarter than me needs to work on it for sure.
Okay, smarter than me too. We're just starting the conversation here. My last question for you. We were talking about anecdotes before. And humans, I always feel humans are natural storytellers. We tell stories. We maybe attribute things to things that happened in our past, when it comes to causation. But is there a story from your experience as a clinician that helped shape how you think about vaccines or public health more broadly?
Yes, thank you. And before I went into medicine, I wanted to be a writer. I was a creative writing major in college, and then when I went into medicine, I became interested in narrative medicine, which is sort of bridging the two fields and looking at the narrative arc and the importance of storytelling. And I do think that stories are very powerful. That's how we remember things. That's how we share experiences. And yeah, I mean, COVID was just so awful. It was just horrific. And it's so much better now, and it has been for years. Um, 2020, like I said, was just an absolute nightmare. And I mean, everyone realizes that, hopefully. I'm not hopefully, but I'm not the only one who thought it was a nightmare. 2021, though, was a nightmare on a whole nother level because I saw so many people die and suffer, and not just die, but some had, prolonged hospitalizations. But yeah, a number of deaths that were 100% preventable. Um, and I shared this on social media recently, and of course I've got to be careful not to share any personal health information, but um, and I won't. But yeah, I mean, I saw people who had kids that died from severe COVID, and made it clear to me before they got really sick that they were adamantly opposed to vaccination. And yeah, I mean, I wrote a piece that was in Stat News recently that talks about this. I mean, I will honestly just be haunted for a long time about some of those cases. These were people who, were just did not need to die. I mean, nobody needs to die, but with the COVID vaccine being as effective as it was and remains to protect against severe disease, um, I really saw these people as victims of bad information, whatever you want to call it, misinformation. And I don't blame them at all, honestly. But it's just so tragic when someone dies, especially if they're young or if they've got kids, and it was completely preventable by a vaccine. So obviously the infection itself wouldn't have been completely preventable. And that was a huge misunderstanding during COVID. There was this totally false assumption that the vaccines prevented infection. That was completely unrealistic. What they do, and what we should have known was the goal or expectation was they blunt the severity of disease. They protect against life-threatening illness. And I guarantee that if these people had gotten the COVID vaccine, they wouldn't have died. Their kids wouldn't have lost their mom or their dad. And so those I think are the stories that compel me to do what I'm doing. I get a lot of flack online for being a pharma shill. I'm not. I haven't gotten a dime from pharma, but the assumption is always that, oh, he's pro-vaccine because he's getting paid to say this or do this. No, this is entirely. I mean, this interview is, I'm volunteering to do this. The stuff I do with my data collection, entirely voluntary. The social media posting, which can take a lot of time, is entirely voluntary. And that's because I don't want people to lose a family member from a totally vaccine preventable illness. And I don't want someone to die from a vaccine preventable illness. So.
All right, and you are on Twitter. I follow you, or X. Sorry,
I
X.
still follow Twitter.
And you
and Blue Sky. What's that?
Oh, you're on Blue Sky. I joined Blue Sky, but it's not. I feel like it's not very active. But
yeah,
okay.
Yeah,
Okay.
they're different, but
They are different.
yeah.
They are different. But thank you so much for this. I just wanted to ask you, go back, because you just sort of made me think of this. When we say, and when we are promoting a vaccine, and like we kind of use the simplified language on purpose, maybe to reach as many people as possible, and we say safe and effective. I'm, in that dialogue, probably backfired during COVID. And I'm wondering if there's room for more nuanced conversation.
Yes.
Yeah, something that we're just not getting right. And then we end up pissing people off.
Totally.
They're like, you said safe and effective, and then they absolutely have like a point, you know, and then you're like, well, yeah, so what? Okay, go ahead. What are your thoughts?
100%. You asked such great questions. I mean, a big focus for me on social media during COVID was explaining the nuances, which is hard. Like, people don't have time and limited understanding when it comes to the nuances. But yeah, safe and effective, that is problematic because there's no such thing as safe. Nothing is 100% safe. Ibuprofen, Tylenol, vitamins, walking across the street, none of that stuff is 100% safe. It's all about a spectrum and putting it into context. When it comes to life-threatening and rare complications, yeah, these medications are safe, but nothing is 100% safe. And then effective. If we say the COVID vaccine is effective and someone gets the COVID vaccine and then 2 months later they get COVID, they think, oh, that wasn't effective. I got COVID. I often hear that with the flu vaccine. I don't get the flu vaccine because I got the flu vaccine once and then I got the flu. Well, that's not what the flu vaccine does. It doesn't prevent infection, and neither does the COVID vaccine. It might lower your risk of getting infected a bit, but it's certainly not 100% or even 50% effective at that. But they're very effective at keeping you alive, keeping you out of the ICU. And so those are the nuances that were completely lost in large-scale science communication during COVID. And yes, there's a lot of room for improvement on that. So we need to give people the benefit of the doubt that they want nuanced information and they want context. I mean, that's one thing that's so encouraging about my recent post that was clarifying a lot of misconceptions. That so many people engage with it, and so many people ask great questions about adjuvants and this and that, and people want to be informed, and people are well informed, and we have to respect that.
Yeah.
So.
Yeah. All right. Well, I want to thank our viewers for joining in, and thank you, Dr. Scott, so much. This was great, very informative. And for our viewers, if they have any comments or questions, post below, and maybe Dr. Scott will let me email him these questions, and I can pass them along, or feel free to come back on. I think I saw on your Twitter that you were open to debates. I thought that was interesting. Did I? Maybe I didn't. Maybe it was.
Yeah, no, sure. I am
You
because
are? Okay.
again, I just want people to know the best information, and I'm not saying I have the best information, but I've put a lot of time into this.
Yeah. Okay.
A lot of time, and I have firsthand experience seeing patients.
Yeah.
So, yeah, I mean, I don't claim to be a world expert, but I'd be happy.
No one
I'd be
knows
happy
everything,
to
but you
have a conversation. And I'm not a confrontational person. I'm not a debater, but I'll do whatever it takes to try to sort of clear the record and make sure people understand what the facts really
debates
say.
can be hit or miss, like you could be walking into a hornet's nest, or it could be like a great place to exchange ideas.
Yeah, honestly, at this point in my life, I don't even care. Like I'll do it. Like I have nothing to lose. I'll walk into debate with anybody about this stuff.
Okay.
And not because I'm cocky, because I know it sounds like that, but I'm not.
No.
I just I just feel like the stakes are so high, and I know the information because I live and breathe it, and I'd be happy to try to clear the air.
All right. You can take a hit and get back up again.
Yeah.
There you go.
Yeah.
All right. Well, thanks so much for your time. And to our viewers, definitely check out trialsitenews.com for more articles. They cover the news, and there's opinion pieces there. And so don't forget to subscribe to the podcast. Thank you.